Hello BIO178 and welcome to the module overview for our Genomics and Transcriptomics module. So we've been talking about high throughput sequencing throughout the semester, but we've been focusing more on the genomic side. So what we're going to do in this module is first of all delve a little bit deeper into the genomics in terms of what the actual workflow is, and then we're also going to focus on this other type of high throughput sequencing called transcriptomics, and with that approach what we're doing is using high throughput sequencing, but instead of looking at the sequence of genes for SNPs or something like that, we're actually looking at the expression level of different genes. So an example of that in a way relates to our everyday lives. This is a paper I found from 2018 where they were using this transcriptomics approach looking at expression levels to look specifically at the effect of anticoagulant poisons in bobcats in Southern California. So this is actually something I had never considered before, but a lot of people use these anticoagulant poisons for rodents, rats. What happens is those bobcats then eat those rats and essentially become secondarily poisoned. It's not lethal, so the bobcats end up surviving, but what this paper was looking at was some of the sublethal gene expression effects of these poisons on the bobcats. So they were able to find some effects in terms of immune function. What else did they find? I'm looking at my notes. Leukocyte differentiation. And so really this is a good example of what you can do with these types of studies. What's cool about this transcriptomics approach is instead of having to look at target genes, so in this study, instead of having to say, okay, we want to look at gene X, Y, and Z and their expression level in these bobcats in response to the poisons, this high throughput sequencing transcriptomics approach allows you to look at every single gene that's expressed in these bobcats. And so it gives a much more complete picture of how they're responding system-wide to these poisons that are in their environment. So for this module, there are a couple different things you'll be able to do by the time you finish the module. First of all, you'll be able to describe the overall workflow and analysis of genomics and transcriptomic studies. So we'll dig a little bit deeper there. You'll be able to explain an RNA-seq experiment. RNA-seq is one type of transcriptomic study. It's the most common one. You'll be able to explain an RNA-seq experiment you could perform to identify genes underlying ecological divergence, or again, that's often synonymous with local adaptation. You'll be able to indicate whether you would need to perform genome sequencing, transcriptome sequencing, or either to accomplish a given research goal. And then lastly, like we've been doing, you'll be able to choose a scientific paper that could be used to write a proposal related to genomics or transcriptomics. So there are a couple main assignments in addition to the traditional mini quizzes I have after each video. One of them is this summary assignment where, like I said, by the end of this module, you'll be able to differentiate, given a specific research question or scenario, whether you would need to use genomic sequencing or transcriptomic sequencing, or you could use either to accomplish that research goal. This is similar to some of the summary quizzes, quote-unquote quizzes we've had at the end of previous modules with molecular markers, for instance, where you were asked to put everything together and determine which molecular marker you would use. This is similar in the sense that I'll give you different scenarios and then ask you to decide which type of high throughput sequencing approach would be most appropriate. And again, just like we've been doing for the last assignment, I'll ask you to find a paper related to genomics or transcriptomics. This is one paper I found that just came out this summer. And what they're doing in this paper is looking at the environmental stress response in Acropora corals, which are shown here. We've talked about these corals a little bit in some previous modules in relation to balancing selection, for instance, and in relation to tying in the SNP allele frequencies with the temperature variation in the environment in American Samoa. That's one example we've used before. In this paper, what they did is they were again looking at the gene expression response of these different corals in response to different types of stress. And they actually found that there were two different general stress type responses in these corals, depending on whether the stress they were exposed to was considered high intensity or low intensity. And of course, this is relevant because many corals worldwide are bleaching and dying because of climate change. That has implications for the ecosystem. Corals are these very important foundation species that provide habitat for tons of other animals on the reef. It's important for tourism, all that kinds of stuff. So this is a cool paper that was looking at what the gene expression level is across all the genes in these corals in response to stress. And of course, I know I sound like a broken record every time I say this, but we're using this information also to build towards writing your proposal. Remember, you're writing that one final proposal at the end. You're writing 2 mini proposals in between. We've already done the first one. For the second mini proposal, if you wanted to incorporate some kind of genomics or transcriptomics approach or aim, you would need the information contained in this module to help you accomplish that.